Now we have like 15 minutes for questions, so I would like to invite all the speakers to come to the front line. And the way we are going to do this is you ask the questions and I will repeat them in the microphone, so everybody can listen them properly, and then we give the question to the person. So if anybody wants to start. Yes? Yeah. Mm-hmm. So Carlota from Spain has a question for you, Sigrid. So if you are taking into account liver function in the way that the drugs are metabolized, to see if that might influence the effect of the treatment, right?
Yeah, so for this, so if I understand the question correctly, so for these treatment therapies that we are including in our drug screen, we know that they don't need to be activated by enzymes in the liver. They are not pro-drugs, if that is what she's wondering. So they should be active independently of the liver enzymes. Did I understand it correctly?
Yes.
Yeah.
More questions? Yes, Paula?
So for me, it's both for Sigrid and Vicente. It's about personalized medicine. It's supposed to be the future, and some cases they could say it's already here. But when we talk about health economics, how should we fit that in? Is really going to be approved by the hospitals because it's economically sensible or not?
Yeah. So Paula,
yeah, I could hear the.
Anyway,
Yeah, I could hear.
So.
Yeah.
Yes. So the question is for both Sigrid and Vicent to see if the hospitals would implement personalized medicine if it's economically relevant, right?
Yeah, so I can start. So yes, this is a very important question, I think, because there are big differences between countries and the economy, and also advances, access to therapies. So this we have included in our EU project. We have a group, health economics group, that will work on these questions to evaluate if it's really feasible to implement this in the health system in different countries, like low income and high income countries.
And if I want to just to add something, it's I think it depends a lot on who pays in each country, like insurance-based countries or let's say more European-style welfare state countries. Who pays? That's important to define that, and also we who are more, let's say, more the molecular biology sometimes are not seeing patients. I think Sigrid is in a nice spot because she's in between, so that's really nice. But sometimes it's more very difficult to see all this extremely good information that we have to translate that to the clinician to be able to use it. So we have to work a lot into communicate with each other, and that's quite a lot of work.
Oh yeah, yes, sorry.
This is a question for Professor Mujica. Actually, I would like you to talk or to elaborate on the challenges that CRISPR-Cas system may have when targeting diseases. Let me give an example that I can think of. Most often one can think of targeting cancer, but if I would like to target like disorders in the CNS, in the central nervous system, for example, right, where cells are not dividing, is that a challenge to think of? I'm guessing yes, and if yes, could you think of ways to overcome these challenges?
So let me repeat the question. Is for Professor Mojica. So it's how can CRISPR technology face challenges when treating diseases, like for example in cells like in the nervous system that are not actively dividing?
Okay, the short answer is I have no idea, because I'm microbiologist, and I don't even use the CRISPR technology, but I can tell you from my point of view of a microbiologist that there are many approaches that you can use just thanks to the vast diversity of CRISPR-Cas systems, and mainly the delivery systems that people is deploying, such nanoparticles and things like that. I talking by her, I remember a few things very recent related to that. I know that people is concerned about delivery. That's a very important point. Accuracy, okay, because this is a system that the prokaryotes invented 3,000 million years ago to fight against viruses. And what they just try is to destroy something that is similar to the memory they got from that virus, because bacteria know that viruses evolve very fast. So they have to tolerate changes. So that means that if they took a picture of a virus with blue eyes, and the virus has lentils, it looks like green eyes, still recognize that guy. So that works in nature as an immune system, but that's not very good to recognize just a sequence in the genome of a eukaryotic cell, 3,000 million base pairs, okay, 6 if you consider the 2 copies of the chromosomes. There are many challenges indeed. Other is related to the immunity that many people has against the Cas9 that everyone uses, which derives from a pathogenic organism that is Streptococcus pyogenes, that can be solved. Most of the problems are being solved thanks to the collaboration of many groups, 20 papers every day on CRISPR. Means that there's so many people working on CRISPR that they are improving every single problem they have is going to be solved. I had no idea about the particular problem of the brain, but I guess could be solved sooner or later.
Yes, I have another question for Dr. Mujica.
Mm-hmm.
And it's about the mitochondria that we have inside our human cells and that resemble some prokaryotic organisms. Do they also have a Cas CRISPR system? Or
Yes, so I repeat the question. The mitochondria in the eukaryote cells come from prokaryotics. So do the mitochondria have a CRISPR-Cas system?
I can give you a fantastic answer to that. In 2000, we found regularly spaced repeats in 20 genomes of prokaryotes, most of them in archaea, but also in bacteria, of course. And the titles is something like family of regularly interspaced repeats in bacteria, archaea, and mitochondria. You read that paper? Okay. So Vicia fava, the common bean, has regularly interspaced repeats in plasmids, small plasmids, 3 kb plasmids. They still have that there. They don't have Cas homologs. There are not Cas genes, not any Cas homolog in any eukaryotic cell. So this is there. We don't know why. So it's still there. And it's in the mitochondria of the, no, it's not in mitochondria, it's in, yes, it's in mitochondria. Okay, but the chloroplasts probably were the ones who had those sequences, because the sequences are almost identical to cyanobacteria. The chloroplasts come from an ancestor of cyanobacteria, and the mitochondria comes from an ancestor of rickettsia that usually don't have CRISPR. But anyway, I remember when we published that, 1 year later someone came to me and said, why you say these are CRISPR, these are regularly spaced repeats, because sometimes they are not regularly interspaced. I said, oh, come on. 2,000 million years, and they are still there. But the sequence is 90% identical to those of cyanobacteria, because they come from there. Which is the role they play? I can guess, but I'm wrong many times. But I probably is doing some sort of structural role. They are playing some structural role. Regular linear space repeats. Palindromic. That means they make secondary structures. Probably is for binding some protein or for keeping some structure. Who knows? That is a mystery. Only in this mitochondria of Pythia fava have been found in eukaryotic cells. Good question, thank you.
Yes.
It's me again. This time for Dr. Pellicano. And I have this curiosity. How likely is it that the RNA that you are studying could be delivered by exosomes? So in reality, this is not a product, but something that the cell has been taken out.
So
So I have to repeat, but I don't know how to repeat that question.
What are the possibilities that the cell, as in the RNA that Dr. Pellicano is studying, could be taking up like the exosome to exosome instead of the cell translating this DNA and producing this
Okay.
RNA?
Yeah. Yes. So the question is how the cells that Dr. Pellicano is studying can is the difference in between the cells translating exosome to exosome instead of yes, sorry, I don't know what's happening here. Here, better. So the translation exosome to exosome instead of using the degradation system, right? Yeah.
So yeah, that's as you were pointing, really, we know that there is some traffic of exosome that are these kind of small vesicles that cells can exchange and contain RNA or proteins, especially small RNAs. So we know that can happen. And the quick answer is that we don't know for sure. I could see some experiments where we could prove it. Just by metabolic labeling some cells and mixing with others, and we have done some work in metabolic labeling of RNA to follow the life of the RNA, how they disappear. But in terms of abundance, I think it will be very, very unlikely it's the case, because we know it's exactly the sequence of the gene, and we can get this information for every single gene, so we can really identify that this comes from the cell. Another thing that in our case, what we do that is good and bad, the first thing that we do is we just put all the cells together and then just use phenol and break everything and mix it all together. So we lose all this special information. So we really don't care, but it's a good to keep in mind. So yeah, I think it's something good to keep in mind. Thanks.
So we have time for one or two more questions, I think. Yes.
As an outsider on this topic, Professor Mujica, but perhaps all of them, are there some regulations about using CRISPR? It has such a lot of promise, but obviously to cure disease, but other things can be used as well. So I don't know if they can comment.
Mm-hmm. So a comment about the regulations of using CRISPR systems.
There are some regulations and limits, of course, but they were before CRISPR. And when in 2012, 2013, was like the wolf is coming, the wolf is coming. So you have to change all these rules, these laws, because now you can do things that could be done before, but they took so much time. They were so difficult that no one was worried about that. So now almost anyone can do CRISPR, can CRISPRize cells. So this is an issue that has to be solved, but it has not been completely solved right now. Now there are many submits people is talking about. We have to, to do. We have to agree to reach an agreement, and there are limitations, but they are not enough for that, and probably will take a while, still a while. But this is going so fast that I don't know what they are thinking about. There are some regulations mainly about agriculture, the use of CRISPR in agriculture. Europe said no, we don't want to approve genetically modified organisms, and they put in the same box anything that is modified, doesn't matter how. States gave green light to any organism, that any crop, any plant that has been modified with CRISPR, Australia, Japan, etc. But that will happen probably also with diseases. Not all countries are going to have exactly the same rules. Well, it's a very tough issue, yeah. Welcome.
Okay, we ran out of time, so I want to thank to all of our speakers now. And in 5 minutes we have the next session of sustainability, so see you in 5 minutes. Thank you very much.